Welcome. Thank you so much for joining. Apologies that we're a little bit late. We had some technical difficulties. I'm Alice Kane. I'm an assistant professor here at ISB. And thank you so much for joining the Research Roundtable today. ISB is a nonprofit biomedical research organization located in South Lake Union in Seattle. And we work on some of the most pressing issues in human health, including aging and brain health. And I'll talk to you about aging today, as well as COVID, tuberculosis, and other infectious diseases. We have a big focus on the human microbiome. Yes, as I'll talk about more today, frailty and women's health. ISB conducts our important research thanks in part to the generosity of donors like yourselves. And because ISB is small and flexible, your contributions go further here. And if you're compelled to make a gift of any size to show us that you value what you're going to hear today, please do so at isbscience.org/donate. And a huge thank you to those of you who already support us. For those of you who are new to the quarterly Research Roundtable series, this is a free virtual platform where you can hear from ISB scientists on their latest research, and you can directly ask questions. You can submit a question at any time using the Q&A feature on your screen, and then after the presentation, I hopefully can answer some of them. So, as I said, I'm Alice Kane, an assistant professor at ISB. I started about 3 years ago, and our lab is really focused on finding the biological drivers of frailty and aging. We use a combination of physiological, molecular, and computational approaches to try and better understand how our health changes over time. I did my PhD at the University of Sydney, and then I spent the first period of my postdoc training at Dalhousie University in Canada before a second postdoc position at Harvard Medical School in the lab of David Sinclair. I'm supported by a number of generous funding donations as well as grants that I'll acknowledge at the end. And you'll see a little bit about what our lab is focused on today, including biomarkers of frailty in both men and women, moving into looking at menopause, and trying to understand the core mechanisms of how our bodies age. Again, thank you so much for joining, and I hope that you're able to learn something interesting today. So the title of my talk is How the Body Ages, providing some insights into frailty and women's health across the menopause transition. So just a little bit of an overview. Today, I'm going to talk to you about I'm going to give some background to put into context. So I'll be talking to you about the concept of geroscience, then a little bit about how frailty is assessed in both humans and mouse models, and especially highlighting sex differences in frailty. And then I'll jump into some results from 3 projects in the lab. The first looking at novel frailty biomarkers in mice, the second, novel frailty biomarkers in humans, and then the third, a newer focus of the lab and sort of a pilot project looking at markers of female reproductive aging. So I just want to start with the geroscience hypothesis, and this is basically a big focus of the aging field in the last 10 years or so. And this is the hypothesis that there are core mechanisms that cause aging and that underlie the development of basically all age-related diseases. And so here, this figure here basically shows that as age increases, there's a huge amount of increase in the risk and the poor outcomes, including mortality, related with the huge range of diseases, the most common diseases that we see in aging. So heart disease, cancer, pneumonia, diabetes, Alzheimer's, kidney disease, where the major risk factor for the prevalence and the outcomes of these diseases is aging. And the hypothesis is that if we can target the core mechanisms that cause that change in aging and that cause this increased risk across these diseases, we can delay the full range of these age-related diseases rather than just targeting them individually. And the goal with this is to basically increase what we call our health span. So this trajectory at the top here basically shows what normal aging today looks like. People have a period of relatively good health that we often call their health span, where they maintain independence, function, remain free of chronic disease. And then there's a period of decline later in life where we lose some of this function and often have one or even more chronic diseases. And the goal is to basically boost this health span period, boost this period of health, independence, and function with a smaller decline at the end of life. And although we have a good understanding of what happens as we age, so this is a figure that comes from ISB and basically shows how we can study the concept of systems biology, and here we can apply it beautifully to aging. We know what happens as we age, so we basically lose function and we have a decline in how everything is working across basically any level that we want to assess. So if we look at the level of genes, we have increased gene instability and increased mutations. When we look at the level of molecular networks, and this is something I'll particularly focus on today, we see changes and declines in how these networks are communicating with each other, but also the specific molecules that make up the networks. And then we see also decline in cells at the level of organs. We see functional decline, reduced lung capacity. And this all results in the level of the individual. We see reduced function, and we can assess this with things like gait speed that we know declines with age. And then importantly, this translates even further into our social networks and at the level of community, where basically as we age, we see a breakdown in our social connections and we see a decline in our health at this level as well. So really across the level from gene to society, we see a decline in function as we age. And we know that this happens, but what we don't know are those underlying core mechanisms that cause this aging. And I would argue that one of the main reasons that we don't have a good understanding of those core mechanisms is that it is a non-trivial manner to measure or quantify aging. And so here we're seeing basically a curve that shows, as I was just describing, a period of relatively good health, and the y-axis here can really be any of those functions, those items I was just talking about, looking at cellular function or function at the level of a whole person. A period of good health followed by a period of decline. But there's a huge amount of biological variability in this decline, such that some people maintain their health and function much longer than others. And what this means is that chronological age, or just the passing of time, is not a good measure of health. So if we draw a line here at, say, 80 years of age, we see that there's a huge amount of variability in health for people at the same chronological age. And we have people that are up the top here of this curve that have maintained their function, their independence, and those that have not at the bottom level of the curve here. And we definitely see this in humans, this huge amount of variability. And I think everyone has examples in their lives of people that they know that are aging with these two different trajectories and everywhere along the curve in between. But interestingly, even in the lab, and so we use a lot of mouse models, mice only live for about 3 years. So they provide us a really nice way to be able to study the entire lifespan in just a couple of years. And even in these mouse models that are often even just clones of each other, they're genetically identical, they live in exactly the same environments in the lab, we still see a huge amount of variability in health as they're aging. So we get mice, now this black line, instead of representing 80 years, might represent about 2 years for a mouse. And we still see this variability such that some mice maintain their function, their appearance, and others do not. And so a key focus of the lab is trying to figure out what drives this variability. And in order to do that, we need to have a way to quantify this variability. And so what I would propose is that the best measure of this, of health, variability in health and aging, is something called frailty. And this is a concept that's widely used in the clinical and research literature. And there are several different ways of assessing frailty. But basically, the way that we focus on frailty in the lab is by using something called a frailty index. And this is basically a way of counting health deficits that someone has accumulated over their lifespan. And obviously, there's a lot of words on this slide, but the main takeaway is just that these items include a lot of things. So we usually focus on 30 or more items and across a range of factors. And so these could include comorbidities or other diseases that have been diagnosed. They can include difficulty in what we call activities of daily living or function. So how well somebody can dress themselves or get in and out of bed. And we also include factors like self-reported health, how many medications you're taking, abnormal lab values, as well as symptoms. And so we basically count up the number of deficits that a person has and divide by how many we've measured. And this gives us what we call a frailty index score, where a high score indicates that someone is frail and a low score that they're not. And these indices have been studied extensively and are known to predict a whole range of adverse outcomes. And so this is just some data that I'm going to show you from a study we did about 10 years ago using a large American cohort that have been well studied. And what we have here is a survival curve. So basically your probability of survival is on the y-axis and then over time on the x-axis. And in this first graph, what we've done is stratify people by whether or not they have the metabolic syndrome. And this is basically just a collection of risk factors for cardiovascular or metabolic adverse outcomes. So it includes things like waist circumference, blood pressure, triglycerides. And basically, we see that in this population, at least, there's almost no stratification of survival risk based on the metabolic syndrome or not. These two curves are basically overlaid on top of each other. But if we look at the frailty index, and so here we've, for the same population, now calculated frailty index scores, we see a beautiful stratification of risk. So those with the lowest frailty index scores are up here with the black curve, and then we come through all the way down to those the lowest scores with the black all the way through to the highest scores in pink, who have the lowest survival probability or the highest mortality risk. And this has been shown in a range of other contexts and in other populations where frailty is one of the best predictors of mortality risk, as well as other adverse outcomes, including hospitalization, institutionalization, and other disease risks. Importantly, we're also able to measure frailty in mice. So Susan Howlett's lab at Dalhousie University, where I did some of my training, reverse translated the idea of a frailty index from humans. So basically applied the same approach that we use in humans, but now to mice, so that we can quantify health-related deficits in our mouse models in the lab and really study the frailty process in these models that we can really control. And so in the mice, we are obviously focused on slightly different deficits, things that change in aging with mice and are relevant to their function and outcomes. And there are 31 of these deficits. And in the mice, these include things like vision loss, changes in fur quality, and an example here that I'm going to show this video of that hopefully is working is tail stiffening. So what I want you to focus on is this young mouse. We see that the tail was curving around the Sharpie as we would expect, kind of like a possum. And here we have an old mouse where the tail has almost no responsiveness to the Sharpie. So I'm just going to play it again so we can see the young mouse in comparison. So this beautiful responsiveness of the tail. And then in the old mouse, we see what we call tail stiffening, where the tail is no longer responsive to a stimulus like this. So this old mouse would receive a high score for this item and the young mouse a low score. And we have 30 more similar items that we can assess in the mice. And importantly, this is a fast assessment and it's non-invasive. We're not doing anything that causes the animals discomfort or harm or pain. So it can be done longitudinally as they're aging. And as in humans, we see that the frailty indices are able to quantify heterogeneity in health. So here's an example of some data from a couple of years ago where we looked at about 100 mice. And here, each line is showing an individual mouse as it ages. And so 21 months of age is sort of older middle age for a mouse, perhaps the equivalent of about 60 years old, and then all the way through to the end of life. And we can see that in general, frailty index scores increase as we would expect, but there also is a large amount of variability in this process, and not all the trajectories are the same. Not all mice are accumulating deficits at the same rate, and there's a little bit of noise in this in these trajectories. So really showing that we are able to capture the varied health trajectories that happen with these frailty index scores. And importantly, like in humans, in mice, the frailty index is also predictive of adverse outcomes. So this is looking in an Alzheimer's disease mouse model in particular, and it shows a very similar survival curve to what we saw in our human data, but now the time to death is in days rather than years. And we basically see that those mice that have the highest frailty index scores, shown with the dashed line here, have a lower chance of survival as they age than those with the lowest scores. So just to show the main takeaway that the frailty indices, even in mice, provide us a really good way of quantifying aging. Excuse me. And I just importantly want to highlight one of the things that's most interesting about the frailty index, which are that there are very clear sex differences, and these are shown consistently across populations. And this is just an example from a study that was published a few years ago, again from an American cohort, where we see that at every single age here, starting from 50 all the way to 90, and we even see it at younger ages, women have higher frailty index scores than men. And this is despite the fact that women have a lower mortality risk. That women have a lower mortality risk than men. So we get this paradox where women are more frail but have a lower mortality risk at almost every age. And this is the same as what we see in our mice. So this is in mouse now, and we can see that as the age increases, frailty increases as we would expect, but that especially at the oldest ages, but really at most ages, the females are still more frail than the males. And the reasons for this are not known at all. And it's a really interesting paradox. And one of the things that we're really excited about exploring in the lab. There's definitely a contribution of socio-behavioral factors. And at the population level, we know that these are different, that some of these factors are different between men and women, and particularly things like reporting symptoms or seeking healthcare are much more common in women than they are in men. So it could just be a bias in the reporting of these deficits or the capturing of accurate information about these deficits in aging that's contributing. And there were also differences in terms of caregiving responsibilities and a number of other things that could explain some of these differences between men and women in aging. There were also almost certainly a contribution of biological differences, and especially as we still see this bias, this sex paradox bias in our mouse studies, where there aren't as strong differences in terms of socio-behavioral factors. And so the chromosomes, obviously the XX versus XY chromosomes could be contributing to this difference that we're seeing. And then the other key biological difference that is obviously affecting the aging process differently in men and women is hormones, and particularly the shift of menopause that we see in women as they age. But the various contributions of these to this paradox are still not understood at all. And so this is basically all of this background leads to what our lab is interested in. So we are basically trying to find biological determinants of frailty in both sexes. And we use mouse models, as I've shown, but then also human data and human cohorts. And we're interested in this for two reasons. Frailty in and of itself is a significant health issue in the aging population, especially for women. And we have no good biomarkers of frailty, and we especially don't have any good validated interventions to either delay frailty or more importantly, to prevent frailty. And frailty, as I've shown, also provides us a really interesting way of basically studying the aging process itself and quantifying aging in order to really understand these core mechanisms that underlie the aging process or the decline in health that we see in aging. And so I'm going to jump into some projects from the lab now. I'm going to be talking about 3 today, and this is the first of those projects, which is focused on biomarkers of frailty in aging mouse models. And so we use male and female. This is the particular strain of mice we use, C57 black 6 mice that we get from the National Institute of Aging. They're black, as you can see here. They look they're actually very cute, and we follow them across their lifespan. So we start this study in particular, we started at 13 months of age, which is about lower middle age here, so about the equivalent of a 40-year-old, I would say. So we started about 13 months of age, the equivalent of about 40 years. And every couple of months, we do a comprehensive frailty assessment, and then we also collect a blood sample. And we follow mice across their lifespan all the way until the natural end of life. And what this allows us to do is match molecules or outcomes that we can assess in these blood samples with the frailty at that exact time point. Excuse me, with the goal of identifying biomarkers that are associated with, or even better, predict frailty. And so what we focused on is a number of different outcomes. This is a systems biology institute, so we're interested in a variety of factors and levels of assessments. But what I'm going to be talking about today and what we started with is metabolomics. And so this is a very complicated map. I've made it fuzzy on purpose so that you're not distracted by the details. But this is basically just a map of the huge range of metabolic processes that can happen in our body. And these are basically the chemical reactions that are constantly happening in our cells, converting molecules into other molecules. And metabolites or metabolomics basically measures the end output of these chemical reactions. And so we can focus in on each of these reactions and the specific metabolites or chemicals, molecules that are being produced. And we can assess those in the plasma. And it basically gives us a snapshot of all the metabolism that is happening. And so we did this with a company. We were able to detect about 600 metabolites and associate them with frailty. And so the first point that I want to show you here is, and I just will highlight now, and again at the end, that this work was done by Dantong Zhu, an incredibly talented postdoc in the lab. And what we were interested in was distinguishing metabolomic biomarkers of aging as opposed to frailty. So we really wanted to know what wasn't just tracking with the progression of time, but what was associating with our frailty index with this health outcome in aging. And so this here just shows some of the pathways, so we can identify these specific chemicals and then group them based on the pathways that they are part of, some of the pathways that are associated with aging itself. And here we see things like fatty acid metabolism, mineral absorption, bile secretion. And then we can do analyses that basically takes out the aging component. And so now we're just looking at what has changed with frailty independent of age. And this is shown on the right-hand side here. And we can see that the pathways that are changed with frailty are completely different from those changed with aging. And there's a long list of things here, but all the ones that I'm outlining in orange here are related to amino acid metabolism. So these are kind of the basic protein building blocks that make up the proteins in our body. And so it does seem like there's a pretty strong amino acid metabolism dysregulation that's happening with frailty in particular, but not with chronological aging. And so this is exciting pathways that we can follow up in terms of looking at mechanisms, but also identifying specific biomarkers. And so if we look then at the specific chemicals that make up these pathways rather than at the systems level, we can look at individual molecules that predict not just current frailty, but future frailty. And so when we did that analysis, that's basically what this is showing here. We see that alpha-ketoglutarate and levels of alpha-ketoglutarate was the only metabolite where, if you measured it at baseline, predicted whether you were going to be frail or not at the next time point. Predicted whether our mice, apologies, were going to be frail or not at the next time point. And alpha-ketoglutarate is really interesting. It's a supplement. It has antioxidant capacity and has been studied by other groups recently. And in fact, there was a mouse study where they provided alpha-ketoglutarate, to their mice in the diet and saw that it delayed frailty and even made the mice live slightly longer. So it does seem like we're seeing that same thing here, where if you have low levels of alpha-ketoglutarate, you are much more likely to increase your frailty over the next time point. So this provides a really interesting and potentially powerful biomarker that could be measured in the blood and tell you your risk of frailty if you're a mouse. We also then were able to look at not just future frailty, but change in frailty. So basically now looking at frailty trajectories compared to your baseline, are you likely to increase or decrease frailty over the next time point? And we were able to identify a few more biomarkers here. We can see creatinine, but the one that I specifically want to highlight here is nicotinamide riboside, which some of you might be familiar. This is NR. This is one of the NAD boosters that are also commonly studied and talked about in the sort of longevity aging field. We know that NAD levels decline as we age, and boosting them with supplements like NR and related metabolites can have some benefit. And so it was interesting here that we saw that lower levels of nicotinamide riboside were associated with an increased risk of increasing frailty over the next time point. So it does provide some evidence that perhaps there is advantage to supplementing these. Okay, and then the other really important factor is, of course, sex differences. So those two slides that I just showed you were looking at the males and females together, what's changing in both sexes. But if we break it down and stratify and separately look at what's associated with frailty in our males and females, these are different. And so here we see the females here. The colors here just represent two different frailty outcomes that we looked at. And we can see that there were not that many biomarkers that were associated with frailty in the females. And the top one here was FAD, which is a vitamin B derivative. So implying perhaps the vitamin B pathway is particularly important for frailty in females. In the males, there were a few more that came up, and this might just be related to sort of the power of our study. We didn't have hundreds of mice. We were looking at about 40 mice per sex. So whether this means anything or not is still to be determined. But the main takeaway here is that the metabolites that are associated with frailty are different in the males compared to the females. These lists don't overlap. And to highlight some of the particularly interesting ones in the males, we have creatine here, where lower levels of creatine were associated with frailty in our male mice only. And you may be familiar with this as the muscle building supplement that bodybuilders and other people who are going to the gym regularly take. And so it does seem like there may be some advantage there, or at least that low levels of creatine are definitely associated with frailty. So there may be some benefit to boosting. And then sphingomyelins are a type of lipid that in other studies, they've also seen declined with aging across both sexes. So interesting that we saw that was particularly important in the males here. Okay, so just to summarize our takeaways from this mouse project, our main findings were really that frailty is distinct from aging, from chronological aging, and characterized by different pathways, and in particular, amino acid metabolism dysregulation. We also identified some specific markers of future frailty or frailty trajectories, and these were particularly interesting were NR and alpha-ketoglutarate. And we also identified some sex-specific frailty biomarkers, in particular B vitamins for females and the sphingomyelins for males. And the mice provide a really powerful model that we can use to study aging. We can control everything about their life and their environment so that we really know that we're only studying the thing that we are interested in. But obviously, the goal is to find biomarkers and mechanisms that predict frailty or apply to frailty in humans. And so that brings me to project 2, which is using cohort data. And this was a hugely collaborative project done with members of the Hood lab, particularly Noah, as well as the Gibbons lab. This is Alex, who works with Sean in the Gibbons lab here at ISB, Hiju, Lance, and then Dantong from my lab. So as well as others. So this was a large collaborative project. But we used cohorts that for which data had already been collected and that we could calculate frailty scores for. And in exactly the same way as the mice, we're then able to look at what molecules in the blood are associated with frailty in people, just in a slightly more heterogeneous population. And so we used 2 cohorts of humans that we could study. The first is the Aravale cohort, which some of the audience might be familiar with. This was run out of ISB, looked at about 3,000 people that were followed for about 18 months. And this was a wellness cohort. So there was an intervention here where people that took part were provided with health coaching and then followed regularly for about 18 months. The other cohort that we have is a much more community-based cohort, and this is the UK Biobank, which is obviously run out of the UK, and they have a much larger cohort, about 500,000 people, but just from the community, no interventions, and they are again followed longitudinally. And so we calculated frailty indices, these measures of health and aging for these 2 populations in order to try and understand what was associated with frailty at the molecular level. And when we look at the frailty trajectories, they're as we expect. So we're seeing frailty here on the y-axis, and then years since first assessment. So basically the years that people have been taking part in the UK Biobank cohort. And over time, frailty increases. And these dotted lines here represent the basically the 95 percentiles of this data. So there's a huge amount of variability in this data. This cohort includes 500,000 people, but in general, frailty indices increase over time, which is exactly what we would expect. And we're also seeing that our females across this entire time span are more frail than our males, which is also exactly what we would expect. Interestingly, in the Araval cohort, we saw the exact opposite trend. And so now we have days since first draw, which is just days since their enrollment in the study, because they were followed for a much shorter period of time. But over this time period, we actually saw frailty index scores decrease, which is very unusual, and a testament to the how well the coaching and the wellness intervention worked. These men and women that were part of this study were able to reduce their frailty index scores over this time frame. And again, we're seeing the variability in the data here with these dotted lines shown. And so now that we knew what was happening with the frailty indices, and we were able to then dig into some of the molecular data to see, in the same way we did in the mice, what was predictive of or associated with frailty. And I'm just going to show the UK Biobank data here. You can look for this work. We're going to be submitting it in the next couple of weeks. We're writing it up currently. So there's a lot more that I can talk about here, but I'm just going to focus in on the UK Biobank data. And here we looked at molecular factors, mostly proteins, but some metabolites that we assessed in the blood. And we basically used an unsupervised approach where we provide all of the data to a machine learning algorithm that is able to look at how the individual metabolites are changing with frailty, not with frailty, are changing in our cohort, and group these into modules, which are assigned arbitrarily assigned colors, as we can see on the y-axis here. Then after the fact, we can look at how these modules are associated with frailty. So it basically gives an unbiased initial approach of seeing what molecules are changing together, and then we can look at how these are changed with frailty, adjusting for a variety of cofactors, including age and including genetics. And so this gives us a long list of modules that are all made up of different molecular factors that are associated with frailty. And so I'm just going to highlight some of them. The top associated module here, this brown 4 module, includes molecules that are related to inflammaging, and particularly IL-10. And so this is super interesting, given previous work in frailty has shown that there is a very important inflammatory component that is associated with frailty. And so that's exactly what we're seeing here. This module here is associated with immune senescence, which also is particularly interesting, where we basically see molecules that are associated with an activated but dysfunctional innate immune system. And then on the other end of the curve here, we also see an association with molecules that are associated with growth hormone and insulin signaling. So this module represents a sort of metabolic health that we're seeing dysfunction associated with frailty as well. And so these all provide really exciting hypothesis-driving findings that we can dig into further to see whether these are mechanistically associated with frailty. You'll also see here that we have both males and females shown on this graph. And so I just want to dig into some of the sex differences as well, which we're always really interested in. The main takeaway was really that we didn't see as strong sex differences as I expected that we would. The vast majority of these modules were associated with frailty in both males and females in the same direction. And it was really just the strength of that association that was different. But for some of these, so this round module here, we do see a much stronger association in males than we do in females. And this is associated with the complement and coagulation cascades, which are basically molecules that affect how our blood clots. And then on the other side, we also see this module that's much more associated in females than males is also associated with thrombosis, so clotting, but particularly the inflammatory parts of that process. So some interesting sex differences that are coming out of this large cohort, but not as specific or strong as expected. And that's probably related to the huge amount of heterogeneity that we get in these human community studies. I do then just have one more slide about this project. So we were really interested in molecular features that are associated with frailty, but super important and often not combined with these molecular studies are the socio-behavioral factors that are associated with frailty. And so here we're showing women, and I'll show you men in a minute, where basically the socio-behavioral factors are on the axis here, and we can see that if the dot is on this side, that these factors are contributing to resilience against frailty becoming worse over time, and then over here increased susceptibility. And so these are mostly what we would expect, but still really interesting to see that we are getting such strong relationships. So things like going to the gym, having a higher income, and being satisfied with your life are all contribute to resilience against frailty. And then on the other side of the curve here, we have serious illness, being unable to work, having insomnia, financial difficulties are all associated with increased susceptibility. And we see similar results for the men, slightly different items that are coming out here, but similarly, having good sleep, going to the gym, having a high income are all associated with resilience, and then being unable to work and having financial difficulties are on the susceptibility side here. So really interesting to look at these contributions in combination with our molecular features, and these might represent lifestyle factors that could also provide options for intervention. Okay, so the takeaways from this project are that there are distinct signatures of frailty independent of chronological age, and particularly this inflammatory signature represents a really interesting potential target. There are sex differences specifically related to coagulation and thrombosis, and that socio-behavioral factors also contribute to our frailty trajectories. All right, and then I'm just going to briefly talk about the third project, and this is a much newer approach of the lab, where we're really interested in how men and women age differently, and particularly in how women age and female reproductive system aging. And we know that the reproductive system, especially in women, ages faster than almost every other tissue. And this is obviously associated specifically with menopause, where women see their decline in ovarian function and changing hormones as they age, which is associated with, of course, reduced fertility, but is also with a huge range of other health outcomes. And we can see it's the figure on the right here that some of the symptoms that are associated with menopause, and there's also increased risk of diseases, osteoporosis, cardiac disease, that happen with this reproductive system aging. And we also have seen in studies that there's an association between an earlier age of menopause and a reduced lifespan. And so it does really seem like there's clear overlap between this reproductive aging and systemic aging. And as with aging itself, as with frailty, the determinants of female reproductive aging, and especially the huge amount of variability that we see in this decline, aren't known. And we have very limited biomarkers and limited treatment options. So there's a lot that to learn here, and a lot of potential to make a difference in this area. And what we're really focused on is particularly the lower female genital tract. So this is basically the cervix and the vagina, as opposed to the uterus and the ovaries that make up the upper tract. And the lower female genital tract just represents a really unique and interesting environment. It is accessible, so it provides a really great opportunity to be able to sample longitudinally and non-invasively, as opposed to doing a biopsy of the ovaries. That's a very invasive procedure, but we have ways of sampling the lower female genital tract in a much less invasive way. And it's also exposed to the environment. So you get a really interesting combination of microbiome, immune function, as well as epithelial cells and the cells that make up the individual tissues. So it just represents a really unique and interesting environment to study, that, yes, represents the interaction of hormones, mucus, immune cells, epithelium, and microbiome, and is influenced, obviously, by a huge range of clinical factors. but has not been studied very extensively in terms of how it ages. And so we were really excited to do a pilot project. And again, this was a hugely collaborative project led by Shailene Frost, a postdoc in my lab, and in collaboration with Ashley Theberg, who's in the chemistry department at the University of Washington, and her grad student Ingrid, as well as some collaborators, Whitney Harrington and Heather Jaspin at the Seattle Children's Research Institute. And so Whitney Harrington and her group had used soft discs, which are basically menstrual discs that can be inserted, and they sit just under the cervix here. Are usually used to collect menstrual blood, but Whitney showed that they could be worn when somebody wasn't menstruating to basically collect cervicovaginal fluid, which includes all the various components I just talked about: immune cells, epithelial cells, microbiome. And we could use this to basically our hypothesis was that we could use this type of sampling and this type of samples to study the aging female reproductive tract. And we were particularly interested in them because they can be used by women themselves. They can be self-inserted, self-collected, can be done in the comfort of someone's home. And so we designed a study with Ashley's group where we enrolled 10 women that were a range of ages. We actually used Facebook for our enrollment. We delivered kits to their home. We asked them to collect samples. So we used a blood sample using what's called a Tasso device, which goes on the upper arm and is able to collect a small amount of blood. Again, this can be done. This is self-done by the women that we enrolled, and they stabilize that for shipping, and then collect their cervical vaginal sample as well and stabilize that, and then ship them back to us. And what we focused on analyzing in the specific cells here was the DNA methylation outcomes, and I'll talk about that in a minute. But this is really just a pilot study, so we only had 10 women, and our main question was, can we even do this? Can we use these samples collected at home and shipped to us to study aging? And our first question was, would women want to do this? And so we asked them how easy it was to use the discs. Everybody said that it was easy and viable to do this. And importantly, 100% of women said they would consider using the discs again in another study. So we were really excited about that. We also did some QC then and looked at the quality of the data that we were getting, and specifically the DNA, and found that it was really high quality. So it did seem like these at-home kits were going to be a viable option to use. Then the second sort of big question we had was whether this particular sample collected in this way would tell us anything about aging. We know that the female reproductive system ages compared to other tissues, and here we had blood as our comparison, but we weren't sure whether this specific sample and the cells that we collect here would show us this age acceleration. And how we chose to measure this was using, as I said, DNA methylation. And again, some of you may be familiar with these kind of epigenetic clocks, they're often called, which provide measures of biological age. And basically how they work is that you compare someone's chronological age with their predicted age, or their age based on their epigenome. So there are algorithms that have been developed that look at the methylation, which is just a specific chemical factor that can be added onto your DNA. And if we look at those specific marks, they correlate really strongly with aging, and they change in a really specific way as we age. And so you can look at someone's age based on their DNA methylation compared to their chronological age, and we see a generally positive correlation here. But we can look at how different they are. So this shows a positive residual where someone is predicted to be older than they actually are. And this implies that they're not aging that well, that they have what we call an accelerated biological age, as opposed to the other side of this curve where we see this negative residual, or this person is predicted to be younger than their chronological age. And that means that they're probably doing pretty well. And so we applied these algorithms to both our blood and our CVF samples. And really excitingly, we saw what we hypothesized, which was that these samples would be older than the blood. And so here, this is just showing for each participant in the study, their matched blood sample age. So they were all predicted to be slightly older than their chronological age in their blood, but their CVF was, or their cervicovaginal fluid was predicted to be much older than that. And every person except this one had an older CVF compared to blood. So this was really exciting as kind of the first proof of concept that we could use these samples to study aging. And so their main takeaways were that the kits were viable for the collection of samples across the lifespan, and that the CVF did seem to be accelerated. And so now we're applying for more funding to be able to basically expand this project to really look at changes across menopause and across aging. And we've put in a large grant that actually was reviewed yesterday, so I'm anxiously checking my email to see the result of that. But we're aim to recruit across the US remotely, ship samples out to women, and in premenopause, perimenopause, and postmenopause. And for our first two groups, recollect after 1 year, and they will ship samples back to us. We'll collect extensive health information to be able to look at their frailty as well as other things, and then phenotype not just the DNA outcomes, but also look at the RNA, the immune system, the T cells, the microbiome, the virome, and the other factors here. So stay tuned. In a few years, we hope to have much more information here with this study. Okay, so I'm just going to summarize the overall takeaways. If you get nothing from today, these are the things that I'm hoping you can take away. That frailty in humans and mice provides a way to non-invasively quantify heterogeneity in health, and that using a systems biology approach provides a really powerful framework to study these complex processes of frailty and aging. It's always important to consider sex as a biological variable and to stratify our analyses by sex, and that frailty does seem to be an independent biological state from chronological aging with unique biomarkers and unique mechanisms. And then finally, that studying the aging lower female genital tract may provide insight into biomarkers and treatment strategies for aging women, and where there's more work coming here. Obviously, acknowledgements are super important. This work is all done as part of a huge team. So my lab at ISB, particularly Dan Tong and Shailene, who drove a lot of this work, as well as our collaborators from my postdoc, as well as at ISB, the Lee Hood's lab, and the various collaborators there, as well as the Gibbons lab, who we work closely with, our collaborators at Seattle Children's, and then Ashley Theberg at the University of Washington. And then, of course, my funding. The work cannot be done without funding. So some funding from the Volo Foundation during my postdoc, and then currently supported by the NIA, as well as a very generous donation from Dan and Lee. And I would love to take questions now, I think. Thank you. This one is from Evelyn. She says, in the models where disease onset is pushed out, what does the cause of death pattern look like at, say, 95 to 105? Are people mainly dying of dementia, infections, multi-organ failure, or something else? It's a really great question. So basically, in those sort of theoretical health span figures that I was showing, where we're pushing out someone's health by basically delaying chronic diseases, and then what are they actually dying of? And we don't know, basically, and like that isn't really where we're at yet. So we're not at that point of understanding where, if we push out chronic diseases, what is the actual cause of death? It may well be dementia, it might be falls, it might be things like this, but it's a great question, that if we delay these diseases, what will it be that is actually causing mortality? Okay, and then the next attendee says, given your research, have you changed anything about your lifestyle regarding supplements, training, or anything else? What advice would you give to a loved one to optimize their health span and reduce frailty? Also a great question, and like the obvious thing to ask, of course, and people in my life, of course, want to know the secret. I would say that we're excited about our research to find new targets, but it's early stage, and really the things that we know do the most now, and I think you kind of saw it on that graph with the socio-behavioral things: exercising, eating well, having good social connections, sleeping well. All of these things that we sort of already know are the core things. So I personally tried to improve my sleep massively, and I have two small children, so I work out as much as I can. But really those things that we already know are the main things that I would advise someone right now. There is definitely more to come, and really exciting research happening, but I'm not sure that we're quite there yet with the evidence, at least personally. Okay. Then the next question is a similar approach that I mentioned some supplements that people are experimenting with. Are there natural ways, such as diet or other lifestyle factors, to get the benefits? Are purchasing supplements and other products necessary, or can, say, eating more kale help? Yes, absolutely. So the direct comparison between like the increase that you're going to get in specific metabolites with the supplement versus a diet, I think is not yet known. And this is sort of where some of this research is going. One of the items that I didn't talk about specifically, but that came up as associated with frailty, was this factor ergothioneine, which is in a lot of mushrooms. And so it sort of is thought that maybe eating a bunch of specifically the hen of the woods mushrooms, those ones that we get in the Pacific Northwest, is able to boost this. But the specific comparison of like how many mushrooms you'd have to eat to get the same amount you're going to get from a supplement, we don't really know yet. But as I said in the answer to the other questions, definitely diet and other lifestyle factors, sleeping well, exercising are the best ways to boost these in a natural way. Okay, and then it looks like there's a specific question about a color-coded modules that I might skip because I am not sure exactly which graph we were talking about there. And then another question here says, have you given any thought to how AI might aid or move forward your research? Have any LLMs been pointed at the cohort data and prompted to come up with findings? Yes, absolutely. Great question. And a really exciting way that AI can help us interpret. So for the frailty, the human cohorts data in particular, we didn't people are working on LLMs that specifically focused on health and aging, and pulling out novel hypotheses, novel things that we might have missed connecting the data. So that's coming. How we applied them in particular in that study was to basically contextualize our results. So when we found our list of items that were associated with frailty, we plugged them into LLMs or similar, to basically tell us like, what are these related to? What does this mean? Give us some insight. What are the best interventions to do this? So, and it gave us some interesting context that we then, of course, double check in terms of the literature, double check in terms of making sense with biology and what the variety of experts that I work with know. But yes, absolutely. I feel like these type of the capacity of these AI approaches to really use a large amount of data and give us insight are really important. I do think we have a hard stop at 1 p.m., so I probably can just answer one more question here. I'm just working my way down. The sex differences you found are really interesting, as is the reasons for sex differences are largely unexplained. Do you think this field of study will shed light on sex differences in the near future? Yes, definitely do. And there's been a real push in the last 5 to 10 years that to consider sex. This is a historic problem. Most studies in mice and in humans were done in males. It wasn't really recognized that there was going to be such a big difference. A variety of contextual reasons there that we've not focused as much historically on sex differences and specifically women's health as we should have. And that is being corrected. And there are a number of government as well as private foundations that are putting a lot of money into this. So I do think there's going to be a huge amount of insight that we're going to get in the near future about sex differences and specifically women's health. Absolutely. Okay, I think I will have to stop there. Apologies for all the questions that I didn't get to. I have some closing remarks here. So I hope you enjoyed today's talk. I hope you were able to learn something. Thank you a huge amount for joining today. And just that again to remind that ISB conducts our important research thanks in part to the generosity of donors like you. And if you did find today's discussion valuable, please consider making a gift of any size at isbscience.org/donate. And we greatly appreciate your support. And you can also support us by helping spread the word about our research and events like the Research Roundtable today. Please feel free to talk about ISB and share the content that you've learned today with your networks. And keep an eye on your inbox for future emails about Research Roundtables and other ISB events. Thank you so much for joining us, and thanks for letting me talk about my research. And I hope you have a great rest of your day. Thank you so much.